Thanks, everyone. Well done on coming back for day 2. For those that weren't here yesterday, my name is Claire Stewart. I'm Mito Foundation's Policy and Advocacy Manager, and I'll just be hosting this first session this morning. I'd just like to start the day by acknowledging again the traditional owners of the lands on which we're meeting, the Gadigal people of the Eora Nation, and express my respect and care for Aboriginal people, particularly at the moment after the referendum. I'd also like to acknowledge again all of the people in the room that have been personally impacted by Mito. It's fantastic to have you here, and we know how it can be a huge challenge to get here. It's my pleasure this morning to introduce someone very well known to Mito Foundation and to many of you, Professor David Thorburn. David co-leads, along with John Christodoulou, the MCRI Brain and Mitochondrial Research Group, which is primarily involved in research, but has also been incredibly important, particularly in the diagnosis of children presenting with symptoms of Mito. David's lab has diagnosed more than 700 patients with mitochondrial disease and has a huge number of publications that help to spread that knowledge so that other people around the world can also be diagnosed faster. So his research focuses on the genetic basis of mitochondrial energy generation diseases. So particular interest in understanding how mitochondrial DNA mutations are passed on from mothers to their children and translating this knowledge into approaches for genetic counselling, prenatal diagnosis and prevention. He also is not limited to mitochondrial DNA, but studies the role of nuclear genes. And his lab has identified over 25 novel disease-causing genes that makes an impact around the world to the Mito community. Today, we've asked David to specifically speak about some of the projects that he has run with the support of Mito Foundation, sometimes financial support, but always the support of us as partner organisations, and particularly with the incredibly important support of people with Mito in Australia as research participants. Thanks, David.
Thanks very much, Claire, and thank you for the invitation to speak today. I always find it really rewarding to interact with the Mito Foundation. It's really been a spur for my career and driven me on, and it's amazing to see the impact on our students in being involved in mitochondrial information days and so on. So I'm always really very happy and pleased to take part in these sort of sessions. So yeah, Claire said I'm sort of asked to speak a little bit about some of the research projects we've been involved in that are either supported financially from the Mito Foundation or have had lots of engagement that has informed the way they work. And I should say, so I'm from Murdoch Children's Research Institute and VCGS, the Victorian Clinical Genetic Services, University of Melbourne, and I am also a board member of the Mito Foundation. So I have a few hats there. So I'm going to talk about 3 projects, and I'm going to try to do them fairly quickly, bit of a whirlwind, not go into a deep dive in most of them. I am going to be around at least until 2 or so today, and very happy to talk in more detail about things. So the first one is one that was funded by, by a group called Australian Genomics primarily. So Australian Genomics was funded by a grant from the National Health and Medical Research Council back in 2016 for $25 million, the biggest grant that ever given, which was specifically to implement all these changes we hear about in genomics into the Australian health system. And it's been refunded and is in the process of being, if you like, converting into a government agency, which is dragging on a little. And so the impetus for this was that there's all sorts of research that's been happening in genomics, and research is fantastic. Research makes all the discoveries, but it's no use unless you can implement it, basically. And so, it's one thing to do when you do research, making these tricky diagnoses in a context where you have years to do it and so on, and it's a little bit different trying to offer it to all comers in a better time frame. So, in the past, diagnosis of mitochondrial disease usually started with a muscle biopsy very often, and maybe we got a diagnosis in less than 20% of patients, say 10 years ago. But with these new genomic technologies that have come in, so that we can sequence, these days the entire DNA, including mitochondrial DNA, depends what you read, that different studies have different amounts and there's a lot of variables there, but it's maybe 30 to 50% of the time we get a genetic diagnosis in a patient with a high suspicion of mitochondrial disease. And those are usually in this research context. So when Australian Genomics was funded, it decided to fund some flagship projects in different areas of rare diseases. And John Christodoulou and I led a mitochondrial flagship, which was a national network comprising most, not all, of the key paediatric and adult centres, recruiting patients from 2017 to 2020. And the entry criteria to be eligible were that the patient had presented to the referring clinician, who was part of the team since 2016. There was a little bit of wobble room there. They'd had little, if any, prior genomic testing. And we had this scoring scheme to grade symptoms and investigations so that prior to testing, they were either a probable or definite type, suspicion of mitochondrial disease, called the modified Nijmegen scheme. Half of them had whole genome sequencing, so that's sequencing all your DNA, including the mitochondrial DNA. The other half just sequenced the coding region of the DNA, about 2%, plus the mitochondrial DNA, so that we could compare them, because genome sequencing is really what we've been wanting to do, but it has been quite expensive, and so it hasn't necessarily been, well, it's not available in most centres around the world, whereas exome sequencing is much more common. So we recruited 140 individuals, 85 with onset in childhood, 55 in adulthood, and we had a $650,000 budget from Australian Genomics, and Mito Foundation were kind enough to supplement that so that we could study more patients. Again, I'm not going to go into a deep dive, but just to emphasise, symptoms are called all sorts of different things. There's a standardized terminology for all symptoms in humanity, pretty much, called the Human Phenotype Ontology, HPO. And so we were coding the symptoms of patients into the database to be able to analyze this. And this is a word cloud where the bigger the writing, the more common the feature was. But I guess I'm just showing it to emphasize that of those 140 patients, we had 120 different distinct clinical terms. And you can see, not surprisingly, muscle weakness, exercise intolerance, intellectual disability, global developmental delay, and increased lactate levels are all some of the things that were pretty common. And so cutting to the chase, in those 140 patients in the timeframe, we achieved an overall diagnostic yield of 55%, 48% being meeting the strictest clinical criteria. And basically of those, the pointer works, sort of those, only about 31% we got that diagnosis directly from doing the sequencing, and then that's clear, we can interpret that this is a diagnosis. With many of the others, we had to do some functional work or do reanalysis a couple of years later, because new genes are discovered all the time, and new variants, new specific family-specific variants in particular genes are discovered all the time, and so the knowledge base accumulates. And in 3 patients, 3 adult patients, we were doing all of this on blood. 3 of the patients we had to go to muscle because we couldn't detect the mitochondrial DNA deletions in blood. So the new technology is fantastic, and for most of the small mitochondrial DNA changes, the 1-letter type changes, I think it's very unusual to be likely to miss that in blood, even with mutations that can go down with age. But for the deletions, they can be just completely undetectable. Sometimes you can see them, but it's not robust in blood. And so, again, just to emphasize the challenges of mitochondrial disease, I guess, so that 55% was 77 patients. And it's not like there's one common gene accounting for these. There's 61 different genes, no more than 3 patients with changes in the same gene. So, we're not talking about a muscular dystrophy or other kind of condition, which I think you know about, that is much simpler to look at genetically. The other thing that might surprise some people, because we think of mitochondrial disease as rare diseases and so on, but around about 3% of the time, when you're investigating patients, particularly patients with complex presentations, it turns out it's not one genetic disease, it's 2 genetic diseases. So that can be a little bit confusing for trying to work out what to test. And so we had 3 of those cases, and I've mentioned the deletions. So that's a very quick overview of the Mito flagship project. And that has evolved into a project we call mitoMDT, which is really the mitochondrial diagnostic network for genomics and omics. Although if you're trying to work out the acronym, it doesn't really spell that. It spells multi-disciplinary team. So yeah, I'm cheating a bit. And so this is a national project recruiting children and adults with suspected mitochondrial disease, and we'll be recruiting until early 2025. And the idea of this is not just to do genomics, but to apply new technologies called multi-omics, which I'll show on the next slide, to push that diagnostic rate to over 70%. So this was a call from the Medical Research Future Fund for projects to boost diagnostic rates, to that sort of level. So that's our KPI. And so it's actually a 4-year project, typo there, and involving expertise across Australia with international collaborators. And again, in this project, Sean is a co-investigator on this project. And when we approached the Mito Foundation in advance, we said it'd be really good leverage to say that that the foundation would put in some money to expand the number of patients we could test. And so I guess there's a tenfold multiplier there. That money was only going to come if the grant was successful. And so that has been really helpful to give us a bit more flexibility in patient numbers and in supporting admin and in various things. And so this project incorporates the expertise of 6 leading Australian labs, plus a number of clinical sites, which I'll show. So what is multi-omics? I think most people are used to hearing about DNA. So, these billions of base pairs of letters with maybe 20,000 genes. And, the diagnostic testing is typically sequencing the DNA and trying to interpret it. But it's not always obvious. And so in biology, we talk about this central dogma where the DNA is the blueprint and it codes for individual RNAs for each gene, which are the message. And then those are translated into proteins, sequences of amino acids that fold up in very specific ways, and form proteins that can do things structurally or enzymatically. And so effectively, genomics is what can happen. We read it and we can sometimes make 100% certain predictions, and sometimes it's of uncertain significance. Transcriptomics or RNA sequencing is looking at the RNA and you can, there's 20,000 odd genes. We can detect maybe 6,000 to 10,000 of those in blood or cultured skin fibroblasts or so on, and look at the RNA, which can be super helpful for confirming genes. And then more recently, transcriptomics is entering clinical practice. More recently, proteomics has come on board, where again, we can detect about 6,000 different proteins in fibroblasts or lymphocytes that we can prepare in a few hours from blood. So the clinical and research network includes sites in all states. So Queensland Children's Hospital and the Wesley Hospital, Royal North Shore is, we started with Royal North Shore when Carolyn was there, and she's moved to Neura. North Shore, it looks like it'll also be involved now. Sydney Children's Hospital, Royal Children's Hospital and VCGS in Victoria, Women's and Children's, the Royal Adelaide in South Australia, Royal Perth and Perth Children's Hospital in WA, and then the Mito Foundation and these additional research groups. And actually, I'll just put up the photos. So the lead investigators on this project were Dave Coleman in Queensland, Sean Murray, Carolyn Sue, who's moved from Royal North Shore to Neura, Ryan Davis at the Colling Institute, Mike Ryan at Monash University, Di Stoyanovsky and David Stroud, who both lead groups at the Bio21 Institute of the University of Melbourne, myself looking a bit less hairy and a bit younger, I guess, and Alex Filipovska at the Telethon Kids Institute. So that's the key, the research leads. I haven't put all the clinician names here. And in terms of governance, so at the moment, we are recruiting from Victoria, and Tasmanian patients can also feed into that via the Royal Children's Hospital and the VCGS, from Queensland via the Children's Hospital and the Wesley. In New South Wales, the Sydney Children's Hospital Network, so Westmead and Randwick are approved, and we have applications in process at different stages with Neura and Royal North Shore Hospital. In South Australia, Women's and Children's Hospital, Royal Adelaide are on board, and in WA, likewise, we have applications in process. So in total, this project funds up to 250 patients for these multi-omic studies. And I think Carolyn talked about the MBS rebate yesterday, and so that is a good thing for this project in terms of increasing numbers potentially that can be studied. poses a couple of problems for us in getting that diagnostic rate accurate, but that's a good problem. And so just some of the key findings so far, I guess. So to date, we've had, governance delays and all the, the hacking incidents in Australia have meant regulatory requirements have increased and data transfer agreements are the bane of my life. So to date, we've recruited about 30 patients that are either accepted or under review. And for the first 3 patients to come through, we've been able to find evidence either from the RNA sequencing, the proteomics, or targeted studies that have meant a patient who received an initial clinical diagnosis, say, of variants of uncertain significance in a gene that was thought to be a possible diagnosis, we can push up to basically this is the cause of disease, which has a huge impact because that family now has a diagnosis and can enter clinical trials potentially if they're available, allows reproductive options to be offered and so on. In 2 of our patients, plus another 4 we've found from overseas collaborators, we've identified a novel disease gene already and just finishing off work on that. And one of the really exciting things, David Stroud at Bi21 has led this work. I think one of the most exciting things in genomic testing in the last few years is that, the regular genomic testing typically takes a few months to get a diagnosis because it's expensive, the labs batch things and do it in that sort of way. If you throw money at it, so about $15,000 per patient instead of maybe $3,000, you can do that in 3 days if you work up to it. And so a number of labs overseas and in Australia have developed this acute care genomics program, primarily, starting with neonatal ICU units and so on, so that you get a diagnosis while the patient's still there rather than 3 months later. And that is transformative. It's been funded now in Victoria and 1 or 2 other states. And, it's an amazing program. And we want to do the same for the proteomic testing. So David Stroud, we've developed this protocol. We're collecting samples currently from 400 kids via a Happy Kids project, spinning the blood down to make little lymphocyte pellets and turning those around. And in several cases so far, David's lab's been able to do that in 3 days from sample receipt to getting the proteomic result, which is what we want to move towards. And so he has another grant that is also supported indirectly by the Mito Foundation, having part funded a mass spectrometer there that is trying to move that towards clinical practice. Going a little bit too slowly. So the last one I want to talk about is the mitoHOPE program. And I feel, I'm a little bit of a charlatan speaking about this because I don't lead this project. I'm a sort of a small part and I'm a scientist. And so I may not be able to answer all of the detailed questions about clinical pathways and criteria, but I can give it a go. So this is led by Professor John Carroll from Monash University, and its goal is to introduce mitochondrial donation into Australia. I think there's huge buy-in from Monash University in this. John Carroll's the head of the Biomolecular Discovery Institute there. Mike Ryan, who is another lead on this, is the Pro-Vice Chancellor for Research. So there's encouragement from the top. And I think all the admin people and so on have been involved, have been super, super enthusiastic. And as shown by, we had a bloody long walk on the Mornington Peninsula last week, and there were about 30 people in the mitoHOPE program. And we got well above our goal there, getting 14,000, only beaten by Noah's Ninjas, but no one's going to beat Noah's Ninjas, not in Victoria. Who are the people? So John Carroll from Monash, Becky Robka from Adelaide, Mary Herbert, who led the embryology studies in the UK, in Newcastle, but has now moved to Monash University and retaining the links to Newcastle, research embryologists with huge international reputations about mitochondria in eggs and in development. And so they're the kind of the core of the project really is getting that embryology right, because it's not going to work at all if you can't do this. This is not plug and play sort of stuff. It's been done in one site in the world in Newcastle, and we still don't really have outcomes that are available from that. So they're absolutely critical to it. And then they've had existing collaborations with Monash IVF. And so Dee Zander Fox and Luke Romberts are the head of the clinical embryology and medical director of Monash IVF, and super involved. And so there's going to be a research lab set up and a clinical lab set up with the identical equipment and so on to enable transition and training rapidly. The other people, John Christodoulou and myself, lead the clinical hub delivery model for setting up the genetic counselling and the national clinical network for that entry point. You know, Sean and Claire, who are sitting on the exec. Catherine Mills leads the consumer engagement work, and Roseanne Block is the program manager. And it's one of those little nice quirks. Roseanne worked with me in the late 90s, with me and Henrik Dahl, and published the first study looking at pathogenic mutations in human oocytes. So, you know, a real founder publication in this area, then moved into research management, and now she's back organising this. So really good insight from the admin team. I think I've said all of that. So there are people involved in 50 plus hospitals and institutes around the country, but these are the ones that are leading it. So quick summary of the road to mitochondrial donation. I think it was 2008 that the UK changed their law to allow these this sort of technologies to potentially be used. After a series of reviews and lobbying from the Lilly Foundation in Newcastle, the law was enacted in 2015. Newcastle received a clinical license in 2017, and the first patients approved in 2018. And there is still some, if you like, secrecy about the outcome due to concerns around confidentiality, but it has now been publicly acknowledged that there have been babies, fewer than 5 born to date in the UK. In Australia, really the Mito Foundation started lobbying in about 2016, lobbying with a vengeance, as in a serious sort of context, and that led to a Senate inquiry in 2018, to an NHMRC review in 2019 that reported at the start of 2020, and then amazingly, it still blows my mind that Mito Foundation really could achieve this. Got the law changed to allow Maeve's law enabling mitochondrial donation. There was then a grant opportunity announced, and that was awarded to the mitoHOPE consortium in March this year. Funding model. So I'm not sorted until June, and the funding hasn't really started flowing yet and so on. So we're kind of working without agreements in place at the moment, but there is significant progress. And so I sort of put this slide up from the Bloody Lung walk last weekend. That's symbolic halfway. So I think we're much more than halfway in terms of when the lobbying for when things started and so on, but there is still a significant way to go. So this is a research program, and we're moving towards a clinical trial that the legislation says should run for 10 years before it could come into regular clinical practice. And thank you to the Mito Foundation. Seeing Noah wasn't involved in the Bloody Long Walk this year, I was honoured to steal his bid. Yeah. Claire always says it like it is. So the mitoHOPE program essentially includes a clinical network and a clinical trial to provide evidence on the safety, efficacy, and feasibility of implementing mitochondrial donation. In other words, does it work? And how can we run it out in an appropriate way? With an underpinning research program led by those research embryologists to try to improve the techniques so that we get a higher success rate and decreased risk profile. And then there's an outreach and engagement program. 10 minutes. I'm working on it. And so I'm just going to go quickly through some slides, and we can come back to them really. And so the clinical trial pathway involves a number of steps: so trial design, referral pathway, the IVF donation pathway, and follow-up of children born from this technology. So we can come back to any of these. Or
thank you.
That working okay? Sorry, I'm obviously turning too much, am I? And
no, it's just for
yeah, sorry, yeah, sure. Okay, great. So we can come back to any of this. Again, just trying to touch over the surface. So in terms of progress so far, as I said, this started in June effectively. The funding agreement made it clear that we were actually getting the $15 million that was promised to be distributed in many ways. And so key personnel have all been recruited now, including 3 clinical embryologists at Monash IVF who are going to start training up specifically for mitochondrial donation. Governance has been established. I've actually thrown that slide out, but I can show it if you like. Development of all these things. There's a lot of governance around this. I mean, we lobbied, and I think appropriately, that this had to be done in an appropriately regulated context, and that's a little bit of a rod for our own back, because This week, the ethics applications went into Monash University for the preclinical phase, and that's 30 documents, including a 48-page protocol, and weeks of work from the academics and the program support staff and so on, and then engaging with the Embryo Research Licensing Committee, which oversees this, to start to develop the license applications and so on. I think these, Catherine Mills' team is leading stakeholder engagement, which I think started yesterday here, and we have 6 working groups that are working on defining the exact way we are going to counsel patients, what the eligibility criteria are in a definitive way, pregnancy fitness guidelines, antenatal care, and so on. So just to emphasise, it was really helpful during the writing of this grant that the Mito Foundation generated this Our Voice document, where a number of views were expressed, particularly preference for continuous care to be provided by local care teams as much as possible. So we've certainly tried to include that in this model, and this slide shows, well, the chief investigators in green and the clinical services in purple that are involved in this. So we want to be able to offer patients the ability to either have Zoom type consults or to have consults, as close as possible to where they are. And that involves a very large group of people with different expertise. And then Monash IVF, who is actually delivering the assisted reproductive technology, is very well set up for this sort of program. They have, they're one of the largest IVF providers in Australia, with 23 full service clinics shown in blue. So these are the clinics where you can have ovarian hyperstimulation to boost your eggs, and to have those eggs collected and processed, vitrified or frozen, and sent to Monash IVF in Melbourne in readiness for the mitochondrial donation role. So you get a lot of thought trying to go into minimising the travel and all that sort of stuff. So basically, participants and donors will require one visit to Melbourne for the technique to be formed. They have to be synced, and so that has to be done in one place, and that is at the Monash IVF Cremorne site. And the travel and accommodation as needed for those is budgeted into the mitoHOPE program. Speaking with Claire, she wanted to emphasise just some of the ways patients might move through this program, because it's not going to be one path that every individual will follow. And so I've just put up some scenarios here. So the simple one, if you like, is that the parent or parents request and are approved for mitochondrial donation and proceed through the process to have a healthy child. I think it's my second last slide. The type but in some cases, the type of mito disease will not be eligible for the trial. So this technology is only suitable for mitochondrial DNA, and certainly with childhood onset mitochondrial disease. I think Suzanne Salavelt was talking yesterday, most of the changes are nuclear, so this is not relevant. There are other reproductive options. So the plan is ensuring that patients have bona fide mitochondrial DNA disease, and if not, then they will be guided to other appropriate reproductive options. And it's a key, to me, it's a key win of having mito donation available, will be that families who just haven't been informed about and missed out on opportunities for pre-implantation genetic testing or prenatal testing will get to those pathways. I think Suzanne Salavelt yesterday talked about heteroplasmy levels. So, it's a little bit complex, but the mother's heteroplasmy level is important, and the particular variant, because in some cases they will not be eligible. We will know they will not be approved by the Embryo Research Licensing Committee for mitochondrial donation, because the other reproductive options are regarded as suitable. Some parents will decide after going through this that IVF is not their best option. They might decide that pregnancy is not a healthy option for the mum. The patients or parents are seeing their local genetics team, and all that information is being submitted, and then the central multidisciplinary team has to evaluate this to determine whether it can go to a license application. So in some cases, it could be rejected at that level. Some parents might go through all of this and decide not to consent into the clinical trial. We might put in a license application to the Embryo Research Licensing Committee and they reject it. in some cases, there'll be unsuccessful cycles of ovarian hyperstimulation and going through the process, and parents may not be willing to try again. And if there are 3 or more unsuccessful cycles of IVF, there's likely to be no funding for additional cycles. So there are limitations, and I'm not the best person to talk about age and ovarian reserve and so on, but I can try to deal with that if you want to ask about eligibility. But it's best to put in an email to the website. So basically, there's a lot happening. Developing materials from the working groups is going on at pace with 6 different working groups. The community consultation has been, I think, happening from well before the grant applications and is certainly ongoing. A whole lot of governance stuff, which I said is a good thing if a lot of work. Setting up these multidisciplinary team meetings and submitting individual license applications. And we are planning and hoping to begin recruitment in mid-2024. That, of course, depends on the Embryo Research Licence Committee approving our licenses to do this, so that the embryology team can demonstrate that they are using the technique appropriately. So, yeah, if you're interested to find out more about mitoMDT, there's an email address here. And if you're interested in finding out more about mitoHOPE or registering interest, the best way is to go to the website here and to mention why you're interested, potential participants and donors. And as the things evolve over the coming months, you'll be contacted. So I'm going to stop there because I always speak too long.
You've timed that perfectly.
Not the questions, but I will be here for breaks and so on.
Now it's question time.
Yeah.
No, we're good. Take a seat.
Cool.
You're not off the hook.
Yes.
Yeah. I've only been here for 2 years, but I've already he already knows that this is how it works. I just wanted to quickly reflect on that. So it was fantastic to see how those projects are coming together, and something that we're quite interested in at Mito Foundation is how can we make sure that there's a ripple effect from mitochondrial donation. So we did this amazing thing as a community, but we know that it's not relevant for everyone. How can we make sure that whatever we... Use the other one. All right.
So I'm allowed to stand?
I'll let you know when you're allowed to stand. How can we try and have as many positive benefits? And one of those things that you've heard a lot about yesterday and today... Oh, sorry. One of those things you've heard a lot about yesterday and today is the importance of genetic testing. And I just wanted to link that together before we go for questions, because you might be sitting here saying, well, I know that I've got mito, I present with all the clinical signs, but why is genetic testing so important? And reproductive options is only one of those. We know that the clinical trials, even if they're not a precision medicine like Alan talked about, we know that all of these clinical trials of even medicines, classic medicines, they're requiring a genetic diagnosis. So they know that they're trialing that medicine on someone with mitochondrial disease. So if you're sitting here saying that I or my family member, we don't have a definitive genetic diagnosis yet, I just wanted to reassure you that there's lots of reasons to consider chasing one. And the work that David's doing through mitoMDT is going to increase the chance that you'll get one. So if you're sitting here going, why are we talking so much about diagnosis? That's one of the reasons, and happy to talk more about that. The last thing I'll add is also that why I asked David to go through all those different scenarios that might occur for someone that approaches mitoHOPE is that what's on our minds, Sean and I, as we sit in those meetings with the scientists and try to make sure that the program works and does it quickly, is that's a really complicated and scary set of scenarios of what on earth is going to happen to families that approach the program. And we want to make sure that the program is ready to provide support through all of those scenarios. So we're doing our best to advocate based on what you share with us. But during the rest of today, we still have Budima and Lewis here to listen to you. So if you have some thoughts to share on how can a research program support families through all those different scenarios, we would like to hear from you. So please take the time to go into that room throughout the rest of the day and share your thoughts, or listen out for other things that we send you about opportunities to help us get that right. And now questions, now that I've talked a lot. Pam's got her hand up.
Can I just respond to one thing in there, Claire, and just again about the diagnostic certainty. So in this program, we recruited patients with mostly what we thought was was likely, probable, or definite mitochondrial disease. But in terms of what we found, almost a third of them actually turned out to not have a primary mitochondrial disease. So the challenge with mitochondrial disease is that its symptoms overlap a whole lot of other conditions. And so one of the really important things is when one is testing for mitochondrial disease is to take the blinkers off and look beyond that. And so there are these what some people call mitochondrial mimics, or just conditions that have a spectrum of symptoms that overlaps mitochondrial disease. So a lot of people may be walking around convinced they have mitochondrial disease, but it's actually something else. And again, some of these things have better treatment options than mitochondrial disease in general. So it's just another incentive for thinking about diagnostics. And sorry, Pam.
I want to go back. I want to go back a step. People talk to me about why it takes so long to get a diagnosis. Nobody understands that it wasn't until the 80s that a machine was built by Beckman that could unravel the helix. And it wasn't until the helix was unraveled that people could actually read it and start to identify points along the path of the helix. So we have come a long way, but people don't know about that. And I think what you're doing is absolutely fantastic, even though it's not going to help me.
Thanks, Pam. And technology is just constantly changing, and it has been amazing. The original sequencing, you'd sequence one gene at a time. Yeah, the original sequencing, like from the 80s or so until the early 2000s, basically, you'd sequence one gene at a time, and it would have, some of these genes are huge, so it might be 100 different parts of the gene, and it's just an enormous amount of work, and you're doing one at a time. And now we just sequence all 20,000 and dive in, but we're still discovering new genes, like 10 to 20 new mitochondrial disease genes every year, and it's really important to have these efforts around implementation so that that improves. But it is still frustrating that in most centres it's about a 3-month wait for testing. I think one of the benefits of the MBS item number will make it more affordable for children with mitochondrial disease to have what's called trio testing, where the parents are sequenced at the same time, because that just reduces the analysis time remarkably. And so the turnaround time for that is about 8 weeks, for example. Those are the sort of typical numbers at VCGS, 12 weeks for a regular testing, 8 weeks for a trio.
I've got a 2-part question, I guess. The first one being with the mitoHOPE program, once this starts rolling out, who will be responsible for educating other people, such as genetic centres and GPs, for example, at the first point of your referral? If, for example, you just get a new diagnosis from a genetic centre, will they be educated on this option to be able to refer you on to the next stage? Because we all as a community might know about it, but if you are new to a diagnosis and you don't know what your reproductive options are and you haven't been connected with a specialist, who's going to be responsible to educate the other people to then pass this program on? And then the second part is, once you've enrolled in the program, I guess anybody who's gone through IVF, you would know it's a very emotional roller coaster. Who's responsible for being able to carry that person through that journey, not just the scientific process of it?
Thanks, Shelley. So that's the first one is a really complicated question in terms of education, because lots of efforts go into trying to educate GPs and medical students and so on about mitochondrial disease. But of course, there's also 5,000, 6,000 other rare diseases, and so getting it on people's radar is challenging. And Mito Foundation certainly been involved in a number of those. I think the really big benefit of mitoHOPE, to some degree, the controversy around it creates interest. I saw a comment from the head of ethics at Monash University about, this application really niggled my interest, or whatever. followed by 8 pages of comments. So, people are interested in this, and it is an opportunity that the progress of this will get airplay and provide that opportunity. But there are plans. Mito Foundation for years has tried GP education events and providing resources for GPs, and it's part of the bigger plan. So the engagement group from mitoHOPE is planning further education activities. I can't really speak to that in an informed way. Catherine Mills is leading that work, but the engagement stuff is starting here, and again trying to find the wishes of the community. In terms of support during the process, so certainly there's kind of 2 levels. People are going to be being recruited from multiple specialist sites. And so, these are going to be widespread. Every genetic service in Australia will know about this. And so, it will require referral to a genetic service. But we are going to be trying to get it down to that lower level. So once they get there, not every genetics service in the country is part of it, but basically they'll be able to refer in. There are a lot of centres, as I showed on that map, and so there will be genetic counsellors there that will see the patients and have ongoing interactions. We've been talking about the concept of a mito guide or a chaperone or concierge. So one of the things we discussed is having someone in Melbourne who, once someone is, and we haven't quite sorted out the stage yet, whether they are applying for a license or after the license, to be with them for the journey. So there's a constant contact point for that. That can help with referrals and so on for psychosocial support and so on as well. And so with that team approach, hopefully that will be addressed. And then, of course, the fertility counsellors at Monash IVF, there's one lead person. This isn't going to be a different fertility counsellor every time you call Monash IVF. This is one person leading it, who will be supporting the patients through the journey. So, I think we're very aware of those challenges.
Laura? No? Yes?
And my question, I guess, is when. So you said that in July in 2024, maybe we'll allow a month's grace, that the recruitment will start. How long is it from recruitment to actually implantation?
So I guess by showing that slide with the various pathways through the process, there's not one answer there. And so I said we are still aiming and hoping for recruitment in, starting in June. And this is completely dependent on, Monash IVF satisfying the NHMRC Embryo Research Licensing Committee that we have the techniques working appropriately. So, that is out of our hands, if you like. That'll be the decider. So following recruitment, the initial recruitment is at a genetic counselling session, and there will be a series of sessions. The referral, which will include some of the reproductive advice in terms of starting to think about age and probably the hormone levels, the AMH levels that give an indication of ovarian reserve. But I don't want to suggest there are hard and fast rules around that yet. It's out of my expertise. But so for people who continue through the pathway towards mitochondrial donation, there are a series of appointments. They'll be trying to be done as quickly as possible. We don't know how long the Embryo Research Licence Committee will take to approve applications. So it will be months, I imagine, for each individual person, months as a best option, and possibly as short as 3 months, but I'm really guessing to put a time frame on that because it's really outside my skill set.
Any final questions? Nothing online? Lindsay's just checking. And this Lindsay wants to ask a question. So either way, it's Lindsay.
David, I just want to ask, in relation to the individual mito things that you've listed there, like Pol G, OPA1 and all that, how long is it down the track before mito donation might be able to be for a specific gene like that?
Yeah, so mito donation as such is for any mitochondrial DNA change. So it's relevant to anything with the mitochondrial DNA because you're basically replacing mitochondrial DNA.
Yeah.
I think what you're asking about is other gene targeted therapies that, and so there is work going on in a few conditions about gene therapies for nuclear encoded mitochondrial diseases. It's a bit of a tough ask because a lot of these, most of the gene therapies that have moved into clinical practice are for conditions that typically affect mainly one organ system. So, just vision or muscle or whatever. And so with mito disease, a lot of the time you're potentially needing to target multiple tissues and particularly the brain. But so there are moves to that in that area. To me, the sort of work we do in the treatment space is around stem cell models and pharmacological therapies, because to me there is a lot of promise in some of the preclinical data on individual compounds, and there seems to be a lot of engagement from small pharma, particularly developing compounds and wanting to test them. So I kind of like the idea that your specific diagnosis will guide the specific treatment, because there isn't going to be one silver bullet in terms of a drug treatment. The gene therapy approaches are targeting one gene and they are moving very quickly. But yeah, there's a bunch of challenges when there are so many different genes and none of them are common. In one sense, gene therapy is great for conditions, great for conditions affecting primarily one organ system or one or two. I mean, even something like Friedreich's ataxia, which is, does affect multiple organ systems, but is a very consistent gene. All the patients, it's a one gene, one construct can fix it in theory. So for mitochondrial disease, it's 300 plus different genes.
And 10 to 20 adding this year.
Yeah.
Thank you very much. That was a great future-focused question to finish us on. Thank you, David, for transiting via Sydney today. David's on his way to see his grandchildren in Tamworth this afternoon, so was kind enough to stop off on the way from Melbourne. Thank you very much.